So, starting a little bit with the background. Why meta-analytic approaches, which is the title of my talk? Well, we want to understand the brain organization, obviously, as our major goal. Now, one thing I would like to set out from the beginning. I'm a very much system-level guy. So that means you're not going to hear about individual synapses, no individual neurons, not even individual columns, but rather virtually all of what we do happens on the brain level. So I'm concerned with where something is being processed and how these regions interact. This is purely macro scale. Now, and if you look at, from this perspective, at our attempts to understand brain organization, we do have two fundamental principles. One is regional specialization. So the analysis of regionally specific effects. Now, regionally specific again means brain regions in the scale of several millimeters, usually more around the centimeter scale. And the question there that we ask is, which areas constitute a particular neural system? So if you have a particular task that subjects do in the MRI scanner, then we can analyze the changes in blood flow evoked by that task and then see where in the brain was that located. And that's the fundamental principle of neuroimaging. I come to that back in a few slides. So one of our attempts is to localize brain functions. The other is to investigate functional integration, that is how do different brain areas, and mind you, brain areas, not individual neurons or microcircuits, how do they interact? What are the kind of interregional effects in these neuronal systems? And that's basically brain connectivity. So, as I already said, functional neuroimaging allows the in vivo localization of cognitive, sensory, and motor processes in the human brain. Now, I kind of realized I should have added a specific slide on that, but I'm just going to talk you through. So what happens? Subjects go into the MRI scanner, and then they are presented some task that usually is in the way that they have a visual display in front of them, showing them some form of instruction. For example, they could see faces, or they could see objects they have to memorize. They could be instructed to pay attention to a sudden change, or anything like this. And the subjects then in the scanner have to perform that task. They rate how happy a face is, they answer whether this is the same object that they saw before, they press their left or right index finger as soon as they see an error, anything like this. Really anything you see from standard cognitive psychology experiments. Now, while they're doing that, the MRI machine continuously takes images of the brain. So every 2 seconds there's a whole brain image that is being acquired by the MRI machine. Now, the crucial underlying principle of functional imaging is that if you have a regional activation in the brain, if one brain region responds to the given task, then this will lead to a change in blood flow at the same location. That's what's called neurovascular coupling. And you have a set of neurons. Now, with a set of neurons, I mean millions, billions, not a few individual ones, but large population activation. This leads to a release of vasoactive substances, in particular NO, and then it comes to a dilation of the vessels. So the blood flow increases. And this neurovascular coupling is really the fundamental underpinnings of anything related to imaging. It is also, to be honest, one of the weaknesses we have. Why? Because we cannot measure neuronal activity directly. We only measure the hemodynamic response, the local hemodynamic response to a local increase in neural processing. And the other thing is, processing here does not directly relate to anything like spike rate or firing rate. It's mainly actually local field potential. Yeah? So you have local processing. That local processing leads to a local release in vasoactive substances, and this leads to a change in blood flow. Now, that's great, but unfortunately we can't measure blood flow directly with the MRI. So what do we measure? What we measure is the magnetic properties of the blood. Why should that change? Now, the thing is, in the blood we have hemoglobin, which is the red substance in our red blood cells. And the central atom of that hemoglobin complex is an iron atom. Now things become interesting. We are in a magnet, we're talking about iron. Now the thing is, where the magnetic properties of this iron atom depend on whether oxygen is coupled or not. So oxyhemoglobin, so hemoglobin with an oxygen atom on it, has a different magnetic property than desoxyhemoglobin with no oxygen attached. Now, what happens in the brain in rest? You have a certain inflow in blood, and you have a certain oxygen extraction. There's going to be a certain ratio of oxy to desoxyhemoglobin. What now happens if we increase the blood flow? Now the ratio of oxy to desoxyhemoglobin actually increases. So there's more oxygenated blood in a certain area in the brain if it becomes more active. And this is quite different from what you have, for example, in a muscle. If you work a muscle, then what happens to the local oxygen fraction? It actually decreases, because the muscle uses a lot of oxygen. In the brain, the situation is different. The increase in blood flow is much stronger than the increase in oxygen consumption. Why is that? Because the key reason for increasing the blood flow is not to provide more oxygen. The key reason is to allow sort of washout of substances. So, if we follow that cascade, we give the subject a particular task. This task recruits some particular brain areas that are responsible for doing this task. This increased activation leads to an increase in blood flow. This increase in blood flow leads to an increase in the oxy to desoxyhemoglobin ratio. And this, due to the different magnetic properties of oxy and desoxyhemoglobin, leads to an increase in the MRI signal. Now, in practice, what we see is very straightforward a correlation between task and signal. Remember, we put the subject in the scanner, give them the task, we know when something happened during the time that we scanned them, and we can then just test for correlation between the timing of our task and the signal that we measure in the fMRI, in the EPI images. But it's kind of important to remember that it's this direct or this increase in MRI signal does not reflect the change in neuronal activity, but it reflects the change in oxy to desoxy HP ratio that is caused by increased blood flow, that's caused by increased vasoactive substances, and that is caused ultimately, obviously, by neuronal activation. As some of you may already guess, when functional imaging, functional MRI, was first introduced in the early 90s, people were very skeptical to believe that such an indirect measure can actually really tell you something about the brain. Now, 25 years later, we know that it works very well and very robust. So what you can do then is to localize functional modules, functional responses in the brain. So here you just see some example images, and the logic is always the same. The question each of these studies asks is, where in the brain is there an increased signal indicating an increased processing given this or that task. I'll make a brief stop here. And please be honest, who is unsure about anything that I said and who has not or thinks he may not completely have understood what's going on in functional imaging? Because the rest of the talk is going to be fairly boring if you don't, if you didn't get quite all the basics. Everybody happy with that? Okay, so we'll talk about localization of brain functions. Now, this has become an immensely popular technique over the last 20 years. If you look at systems of functional neuroscience in the 80s, you had a lot of primate work, you had a lot of lesion mapping, you had PET investigations. It was really a broad and wide field. If you look now at functional neuroscience, systems neuroscience, it's probably 95 or 99% all fMRI. This has become the method for localizing anything in terms of functional imaging.
So what is the time resolution of this?
Oops. Somehow the connector here is a little bit shaky, but we get it done. Well, it's a little bit difficult to say, because on one hand, it doesn't directly depend on the resolution of the scans. The scans are usually taken every 2 seconds, plus minus. Now, the thing is that the hemodynamic response function actually is kind of sluggish itself. So if you have a localized isolated stimulus now at T0, then it does take around 3 to 4 seconds for the hemodynamic response to start. It then has its peak somewhere around 6 to 8 seconds after the stimulus, and then it decays and it's really back to baseline after about 20 seconds. This is fairly slow. But you can model that because it's a fairly stereotypic response. So rather than correlating the activity at the time of the stimulus, we convolve the input function, our stimulus function, with that hemodynamic response function. So if we have a stimulus here and there, we don't look for is activation higher here, then and then, but rather we look for correlations with the expected hemodynamic response function. And then if you, let's say, statistically optimize the variance in the design, like jittering different stimuli in terms of the onset, randomizing different orders of stimuli, then probably you get down to an effective resolution of something in the range of 2 seconds with standard MRI methods. If you want to invest more into sort of the temporal optimization, you can do 2 things, and ideally you should do them in combination. One is to use a faster sequence. Now the problem is, if you want to go faster, like take an image every second or every half second, then you either have to reduce the field of view, so 2 seconds give you a whole brain image, 1 second gives you half of the brain. Or you can reduce the spatial resolution. So our standard resolution in terms of the voxel, the image element sizes, is around 3 millimeter isotropic. If you increase the size, you go faster because you have to acquire less. So it's basically a trade-off between temporal speed and spatial resolution. And in addition, I'm just going to do the second part of the answer. So one thing is go faster on the imaging, and the second is to use more advanced modeling. Now, what I've been talking about is using the standard hemodynamic response, which fits good but not perfect. If you, let's say, go the extra mile and use, for example, an integrated estimation scheme where you estimate both the neuronal populations and on a voxel-wise, so image point level, also estimate the hemodynamic response with some biological constraint priors, you can go lower in the effective resolution. So, bottom line, with standard techniques, and which is what most people would use, you're somewhere in the resolution of a few seconds. You can go down probably somewhere to the level of a few hundred milliseconds if you invest a lot of time both on the imaging and on the modeling side, but this is fairly rarely actually done. And this really also tells you something about the technique. We have a spatial resolution of a few millimeters, and we have a temporal resolution of a few seconds. So this is really more about localizing where in the brain something happens, rather than to have the exact high resolution timing. MRI is never going to get to things like MEG and EEG can, where you actually look at high frequency oscillations or anything like this. It's really more a spatial than temporal technique. You have another question?
Yeah, I want to go in the same direction, because what I the day before showed that we see, so to say, changes in neuronal activity correlation structure on the time scale of hundreds of milliseconds. Now, we only observe individual neurons, right?
Mm-hmm.
And a small subset of such an area. Now, I wonder if you could, so to say, focus with your analysis to make first a broad, so to say, recording where you see all the areas, then say, this area is always active, and then focus only on this, and then increase, so to say, the time resolution considerably.
The answer is a clear yes and no. So this is something that we do quite often. There would be a different talk, but there are techniques called, for example, dynamic causal modeling, where you just look at a subset of, let's say, 5, 6, 7 areas, and then you model the neuronal activity and the hemodynamic activity for each region, plus their interactions. Then you don't really look at things on a higher time scale, because your physical time scale is still the same, but given the much more precise modeling that allows you to differentiate neuronal from hemodynamic effects, you come down to an effective resolution of probably a few hundred milliseconds. So, yes. Now there comes the no part. The no part is that dynamic causal modeling and some related techniques have, let's say, had a very interesting time course over the last 10 years. It was very slow to be adapted, and it was a big woohoo, that's great. We know everything about the brain now. And over the last couple of years there has been, let's say, a lot more reservation and caution. The problem is, to do this kind of modeling, you have to have a lot of assumptions, because you're working in a Bayesian framework. You have explicit priors on, let's say, the hemodynamic response function, and that's what has caused sort of even people like me who've been very enthusiastic about these techniques to be more careful. You also have a lot of implicit assumption in your model. And in fact, if you look at the various releases of this dynamic causal modeling software, it does happen that with later, more optimized modeling approaches, you get different results than previously. And in fact, even at times, things that made a lot of sense and that were very plausible with one release are not really replicated with the next one. So, to sum that up, I think that the idea behind these kind of models, when you look, exactly what Sonja said, you first localize broadly where are the things that are of interest to me, and then model their activity timescales, their interactions in much more detail. The basic idea is fantastic, and it's exactly where we want to go, but I think about roughly 10, 13 years later, we are, as a field, are much more cautious with these things than we've been a few years ago. Ultimately, however, I think things will move more in that direction. The other thing that kind of also relates to this gap that we have is a potential combination of fMRI and, for example, EEG, where fMRI could help the really, really crappy resolution of EEG, and EEG could help the fairly bad temporal resolution of fMRI. Well, a little anecdote to that end. When I visited my very first human brain mapping meeting many years ago, there was a whole workshop on that, and everyone was saying, well, within the next, we're basically there. The integration is going to be the standard now. Well, quite a few years later now, there's really just a couple of papers that really use a combination of fMRI and EEG, and in fact I've seen almost no application paper that uses a joint analysis rather than analyzing fMRI and EEG separately. So, but really to set a bit more the background, I think there was another interesting thing I got from the last comment, which is really you have to be aware, and I think we as a HBP have to be aware of the gap that we have also from that mesoscopic level, where you look at neuronal populations, sets of individual neurons, to the macroscopic level. There is the gap between the individual cell, the individual synapse, and a microcircuit or a column or a bunch of neurons, but there's also, I think, a fairly large and also a fairly complicated gap between this level and then the kind of system level that I will be talking about today. Okay, so as I said, function imaging, in particular function MRI, has been the tool for basic and clinical neuroscience, neuroimaging, in the last 2 decades. And it's fair to say that it's been a huge success. I think we know a lot more nowadays about the localization of brain function, the differential localization of processes, about dysfunctional activation processes in various clinical populations. So there's a lot to like about MRI. But it does also have its drawbacks. Now, one of them is the fairly small sample size. Usually when you look at neuroimaging, we now look at sample sizes of something about 40, 50 subjects. This is good in a way, and it's way better than what it was at the beginning, where there were studies with 4 or 5 subjects. It's still not all that much if you compare it to other fields of research, in particular clinical research. And this brings me to another, I think, fairly interesting perspective also regarding the HBP. Are we going to look at the sort of one prototypical brain, or do we really want to capture population variance? Now, neuroimaging, functional neuroimaging has moved more towards the later end. We want to know what's going on in the population. We know there's a high inter-individual variability, so we are now fairly ready to acknowledge that even 50 subjects is not all that much. We ideally would like to have hundreds, thousands of subjects, because that would really allow us to get an idea of the population variance in the individual differences and such. But as it stands, I would argue that the fairly small samples that we investigate using neuroimaging, that we have investigated for a long time, are somewhat of a limitation. The second is that we do have indirect measures of neuronal activity. So that means that the way we analyze the data, we measure the data, can influence our results. Now, this is not something particular to neuroimaging. it's maybe a little bit more acknowledged here, at least nowadays. But the thing is, we have to see that depending on how you do the pre-processing and the analysis of your data, your results can vary. And another aspect that I think is more obvious to people coming from cognitive psychology than maybe to you as an audience is that we publish isolated findings. So suppose you do an fMRI study on a particular working memory task and you get some nice findings. Now, what people do usually is write that up and publish it. It's fair, but look as a comparison to what has been the standard in experimental psychology for many years. You do a main experiment, get a finding. And then you do a second experiment addressing one potential confound. You do a third experiment addressing another confound. You do follow-up experiments 3, 4, 5 to test for different variations of the timing of the stimuli, and so on. So there has been a very rich tradition in a field that we are very close to of not believing your main finding, but rather testing for all sorts of variations in experimental design and setup and so on. Now, given the logistic expenses of fMRI, this basically never happens. So that means if you have decided on some experimental parameters, let's stick with a working memory example. Let's say I've decided I want to use letters as my stimuli, not symbols or pictures or whatever. I use letters and I present a letter every 1.5 seconds rather than every second or every 2 seconds. And I ask the subject to give me a response every time a letter was repeated rather than when it came every second or whatever. Then this is my design, this is my analysis. And that obviously means that we cannot be sure in imaging whether slight variations in the setup could have actually led to different results. Other way around, we don't know if our findings are robust against both sort of different analysis approaches or different experimental setups.
I was hoping you might just give us a couple of sentences about your views on the resting state in the default mode network, because I'm just wondering if people are accumulating large databases, whether it might be easier to do it like that. And
yeah.
There seemed to be a real enthusiasm for the resting state, and then nobody wanted to do it anymore, and it was all fMRI. Now people have gone back to talking about the default mode network. So.
Yeah, well, that could easily be another 1-hour talk. I'm going to try to limit myself to 5 minutes. What is this resting state default mode? Well, first and foremost, a lot of it is mashing together lots of different terms. Now, resting state fMRI means you do fMRI measurements, but you don't give subjects a task. They just lie in the scanner and you scan the brain. Now, what happens if you just lie around? You just let your mind wander. So you think about, that's actually aching a bit back there, and oh, I have to go to the shop later, and what was it yesterday with my girlfriend? And so you basically just engage into some sort of endogenously controlled flow of thought. And the interesting thing is what people then found out is that this actually recruits brain networks, obviously because you're thinking, and it does so in a fairly random, but still on the average level, reliable fashion. So that means you can use this kind of resting state images to investigate functional connectivity, functional networks. That means even if you don't give subjects a task, you can estimate how close two brain regions are coupled with each other. Or you can use techniques like independent component analysis to find sets of regions that are coupled in that endogenously controlled state. The big difference is, what I've been talking about up to now, is task-based fMRI. So there is a experimental controlled task that the subjects engage in with a clearly defined timing. You know when something happened. When did the subject press a button? When did he show a particular image? In the resting state, it's endogenously controlled. It's free flow of thought. And so you cannot look for activations, as in what happened at that particular time point. Why not? Because you don't know what happened at that time point. So you can really just look at which brain regions did something together. And this has become very popular for, I think, two main reasons. One is it's simple. You don't need any big setup. You don't need subjects that cooperate all that well. You don't need subjects that follow your instructions. You don't have to have all these response boxes and simulation devices and so on. All you need is a subject you lay in the scanner, and the only requirement there is lie still. And this has made a lot of people very interested in resting state for clinical application. Because the major problem with task fMRI that we have when we investigate clinical populations is that they just don't do the tasks very well. Personally, I've tried to scan people with schizophrenia, and you have people that do fairly okay for 2 or 3 minutes, and then just don't do anything anymore. That's it. I have enough. Or you have subjects that at some point you see they're pressing like randomly. But even when the stimuli came, it's just like, oh, they're playing video games in the head, I don't know. The other thing, even if you have subjects that want to do the task well, you have performance confounds. Think about people with dementia. They're just not so capable of doing things. They make much more errors. So now any difference you see in brain activation, is that due to pathology, or is it just due to the fact that they are not very good? So then you come into performance matching, which is horribly difficult to do. So this was one of the reasons why many people got interested in resting state, as in, well, it would be so easy. Just put them in a scanner, scan 10 minutes, and you got function information. The other aspect of it's so easy to do is it's also very easy to share. There's lots of data available, and really if you want to sort of have the minimal starting investments into a PhD student, set them on resting state images. If it works, great. If not, you have not invested a lot. You didn't use scanner time, you didn't use subject recruitment time, you didn't need to pay subjects, didn't need to pay for the scanner at times, but it's just like, well, here's some images, do something with it. Task images, you can only analyze really once or maybe twice, because there is the particular task, and you look for the effects of that task on the brain. With resting state, you can basically, well, analyze anything you want in there. So these are the pros on the resting state, and probably the reason why it's so popular. The big downside of resting state imaging is we don't really know what we're imaging there. It's a very ill-constrained task, so to speak. There are lots of ways how you can analyze it. We have become now more and more aware of the fact that maybe a lot of the earlier resting state papers really look at differential motion effects rather than brain effects. What is the role of confounds? What is the role of non-neuronal sources of variation? Blood flow, heartbeat. What is maybe even metabolic aspects rather than activation aspects? So, to sum that bit up, resting state allows your users fMRI in the absence of a task. It allows you to investigate functional connectivity as coupling between brain regions. In the context of resting state, that probably means that they are recruited by similar processes going on. It's easy to acquire, easy to share, easy to analyze. It does have the big disadvantage that we have a lot of known and unknown confounds, and that we cannot be really sure what it actually means. Now, one word on the default mode. The default mode network is a particular set of brain regions that usually increases the activation in the absence of a given task. It was defined in the early 2000s by Marcus Raichle and his group as regions showing a high metabolism at rest. It was later shown that these regions have a higher activity level during rest than if you engage in a particular task. And these are also regions that are strongly coupled with each other in the resting state. Now, resting state and default mode are two completely separate things. Resting state is a particular acquisition technique. Functional connectivity is a particular way of analyzing that. The default mode is a particular set of brain regions that have strong functional connectivity in the resting state. Unfortunately, this is very often just meshed together into one sort of indistinguishable blend. And when people talk about, oh, I want to analyze resting state, they mean the default mode. If they talk about default mode, they actually mean functional connectivity and so on. Yeah, so be educated. Resting state, the setting in which you acquire the data. Functional connectivity means I look for correlations, temporal correlations in my data. Default mode is a set of brain regions. If nothing else, you take that home, I'm actually fairly happy. I know that was brief, but did it roughly answer your question? Okay. I want to come to a last point, which I think actually relates to the previous one, and it is really important when you consider neuroimaging. There usually is an overgeneralization of context-specific findings. Now, what does that mean? Suppose you're interested in working memory in schizophrenia, something a lot of people are interested in. Now, what you usually do is to set up an experiment. And as I said, when I talked about this bit here, you need to do a lot of design choices. To be honest, what do most people do? Ask their older colleagues or friends, do you have a design that I can use? Oh yes, here you can have my presentation script. Oh well, perfect, I just use it. That means you have a certain setting of stimulus, timings, required responses, instructions, and you give it the subject. And then you see a difference, let's say, between the patient and the controls. Now, what will you discuss in the discussion of the manuscript? What will you present at conferences? You're not going to say, I find more activation in a task, in a visual task using verbal material, letters to be precise, with a rather slow timing of 2 seconds, if I contrast this condition with that condition in this particular group of subjects. You talk about hypoactivation of the prefrontal cortex in schizophrenia. And this, now I've made it a little bit, exaggerated only actually a bit. The key thing is here, our imaging findings, neuroimaging findings, fMRI findings, are always completely dependent on the experiment. Yeah, we have concepts like attention, working memory, executive control, motor control, whatever. These are broad concepts. You operationalize them with a very particular experiment. And then the findings you get for that particular contrast, in that particular setting, you again overgeneralize and say, well, this is the broader concept I was originally talking about. And this does lead to some problems. So there's a lot of discussion like, is this or that area involved in cognitive control? This is a cognitive controller, or executive control is actually a prime example. You have a lot of people talking about executive cognitive control, that's anterior cingulate cortex. And you have an equally large group saying, well, no, that's actually inferior frontal junction, inferior frontal gyrus. Probably some more people say, well, it's actually insula, some say IPS. Now, if you look closer, you can actually see that it often really depends on the kind of task. If it's more about error monitoring, so if you use error monitoring as your way to operationalize cognitive control, you usually get indeed anterior cingulate. If you use things like task switching or response inhibition, you're much more likely to get lateral prefrontal cortex and so on. So this is something that really has challenged our field in a way that there's lots of ways you can operationalize a broad construct. Depending how you do it, you will get different activation patterns. Now, if you take all of that together, I think it's fair to say that an individual neuroimaging study doesn't tell you a lot. In fact, it tells you close to nothing. But we have a very nice advantage. We have the advantage that there are a lot of neuroimaging studies. This is a somewhat older estimate by Derfus and Mal. So 5 years ago, they estimated about 8,000 studies. If we look now, they're easily about 16 to 20,000 fMRI studies. So the amount of insight we can gain from a single study is fairly limited. But we do have a wealth of studies available to us. And I think that's our biggest advantage. They all report very standardized results. This is due to the fact that from the very beginning, really, of neuroimaging, people have used stereotaxy coordinates. That means that we have a coordinate system for the brain and standard templates that live in that coordinate system that allow you to describe the location of your findings as X, Y, Z. And from the outset of neuroimaging, that is really what people have done. They report their findings as such tables of X, Y, Z coordinates. And now things become really interesting. Each study has a limited insight that you can draw from it. But you have thousands of studies, and all of them report their results in a very standardized, very easy way as XYZ coordinates. So this is really why I think neuroimaging data is almost perfectly suited for pooling and meta-analysis. And this is really what the remainder of the talk is going to be about. I'm going to introduce you to a few of the things that you can do with these kinds of techniques.
Just one other question. Anatomically, people often think of Brodmann areas, and the difficulty with these things is they're hard to equate with the Brodmann area. Is there anybody working on anything to mesh the two?
No. Well, actually, Brodmann areas as such are, well, completely useless.
Not if you're trying to do rat, monkey, man.
Yeah. Now, the thing what I said is Brodmann areas are completely useless. Why is that? Because Brodmann did his histological investigations in an unknown number of brains, and he summarized his results as a schematic two-dimensional drawing. This is really all we have from Brodmann. It's a schematic drawing of the lateral surface, nothing more. And people have then tried to put that into 3D, into standard spaces, but this is so vague that it's basically useless. Now, there are attempts, and I think we Jülich, Katrin Amundsen, and Karl Zillers have been on the forefront of that for many years, to do histological post-mortem investigations in a group of subjects, usually 10, and then bring these results into standard space so that you can do a direct comparison of functional neuroimaging with histologically defined areas in the same reference space. But these are not Brodmann areas. all we have from Brodmann is that one drawing. We cannot compare that directly with imaging data. But you can compare imaging data with histological data, modern histological data, that's in the same kind of reference space. And this will, in a way, allow comparison between species, but I want to be a bit careful there, because the homology also of the different areas between species is not all that clear. And in fact, if you look at primates, you can see that some of the older views, like if you go from human to old world, to new world, to half monkeys, then they have less and less areas. May actually not be true. I just know personally from the work in that field as a PhD student, if you look at the somatosensory cortex, then there was the view that humans have areas 3A, 3B, 1, 2, 3, and a lot of parietal areas, and monkeys, old world monkeys have most of them. New world monkeys probably don't have an area 2. If you go to half monkeys, they have even less, and so on. But subsequent investigations showed actually, no, they have more differentiated cortices as well. So also the monkey data is kind of in flux, and if you look closer, you see that some of the older ideas that the more you go down the evolutionary tree, the farther you go away from humans, the less differentiated cortices. That's probably not true. So this kind of between species comparison has its own tricks associated with it. Key message though, another take-home message: forget Brodmann areas, because Brodmann areas cannot be compared to imaging. Not if you talk about Brodmann areas, what Brodmann has drawn. But yes, comparison with histological, cytoarchitectonic defined areas is important, but they have to be precise, and that means you have to have postmodern data brought into the same space as the imaging results. Any other questions at that point? Okay, then briefly come to what are the two main ways to do meta-analysis of neuroimaging data. Well, one is, broadly speaking, anything that uses the full data. That is, you take images. There are different ways to do that. Now the great thing is, if you have these images, you can do very good and very precise meta-analysis. The problem is, you don't have the images. Try emailing somebody about their 1999 neuron paper. Could you please send me the raw data for that? Do that once, it's actually quite funny. In fact, even if it's just 2 or 3 years ago, and you ask people for just the Z-maps, the final analysis tools, then you hear things like, oh, that was a student that I don't know where he actually went after his PhD, and oh yeah, I will talk to my postdoc who supervised the intern at that time, and I get back to you. They never do, and so on. So really, what we want, what we would need, is ideally all the raw data, or at least the statistical maps, the whole brain statistical maps from each study. But realistically speaking, this is just not feasible. Now, there are attempts to change that. There are repositories being set up. There are projects that are devoted to collecting this kind of data, OpenFMRI, NeuroVault, and so on. I have severe skepticism, because there have been a lot of projects like this over the last years, 10 years probably. All of them failed graciously. So this is where we would like to go, but I'm fairly certain we can't. So what has become the standard is what we would call coordinate-based meta-analysis. So this is based only on the published maxima coordinates. So we have a very, very sparse representation of the results. Yeah, somebody found that big of a block, and we only have the one p coordinate for the maximum. But the big advantage is we can integrate the entire literature because we don't need the help from the original authors anymore. We just need the papers. The coordinates are in the papers, and we can use that to integrate over any time span, any set of experiments. We don't have that strong bias in there, that we can only get data from people that we know well, and even then it's complicated. Now, this also entails a very peculiar aspect of neuroimaging meta-analysis. Usually meta-analysis in any other field of science are about effect sizes. So, is the pooled effect size significant? Now, to do that we would need the original images to have an effect size for every single voxel in the brain. We don't have that. And hence we can only look for the convergence of coordinates. So the key question that we ask is the convergence greater than expected by chance? We now just talk about a spatial property. How is a spatial convergence? We ignore kind of the height information, the effect size. There is no effect size measure in there, but we only look for spatial convergence. Okay, how does it look? Now we come to the first of a set of approaches that I wanted to show you what you can do with these kind of coordinates. And the first is what is really the traditional neuroimaging meta-analysis. I would call that the where approach. Now, to illustrate that, here I show you the location of activations in 73 experiments on finger tapping. So very simple, just move your fingers. Now if you look at that, I guess you can see two things. One is that these activation foci are scattered fairly much around the brain. So there are a lot of different locations that people report activation for in finger tapping studies. But you also see that there seem to be some hotspots. So our question now really is, where do these converge? And you could say, well, here there are a lot of foci, here, yeah, somewhere here, and I'm not quite sure, but here it looks like there's quite a bit as well. So our task now, in terms of meta-analysis, is, given these foci scattered around the brain, to identify those locations where there is a significant convergence of the reported findings. And the key idea behind the approach that we use, activation likelihood estimation, is that the reported coordinates are not treated as points, but rather as centers of probability distributions. So kind of we model the true location as a 3D Gaussian. And why is that? Because we know that there's a certain spatial uncertainty associated with neuroimaging findings.
Is this also the way how maxima are localized, or for the maxima you really just take the single point where you have maximum of single points? Because it could be very unstable.
Yeah. Well, we take as maximum whatever people reported as the x, y, z coordinates in the paper. This is usually the single maximum point, and I think it's that in the vast majority. Some people also report slightly different coordinates, but if you look at the entire field, I would probably say about 90% of the papers report really the peak coordinate, the maxima. And in the paper you only have the peak, so we take that peak and then basically model this 3D Gaussian around it to reflect the spatial uncertainty associated with it. Now, in the interest of time, I'm not going to go into details of spatial uncertainty estimation. Key message here is, if you have a coordinate in a paper, the maxima somebody found in the study, it's their best estimator of where the true effect is, but given that they have a fairly small, usually fairly small population, and given the effects of different spatial normalizations, designs and so on, there is a certain spatial uncertainty for the localization of that peak. And you can model that by this kind of 3D Gaussian. Now, next step would be to replace each and every of these foci by that respective 3D Gaussian, and then take a union across experiments. So that basically tells us how likely was the location of at least one true focus at that particular voxel. And what you see is, actually quite nicely, that you get now a somewhat better idea of the hotspots, where the things converge, but you also see that a lot of the brain really is covered with values. So the next question is, which of these values are significant? And we can assess significance against the null distribution of spatial independence. What does it mean? Here you have 3 individual experiments with their modeled Gaussians. Now what we do in the actual ALE analysis is to compute the union at a particular voxel. So here, if we look at that voxel, we take this value, and this value. So they are spatially at the same location. Now our null hypothesis says location doesn't matter. There is no spatial convergence. So what we can do is pick voxels at random and compute the ALE values. Just pick them from anywhere in the brain. Because remember, location doesn't matter. And if we do that many times, we get a null distribution of what we would expect these ALE values to look like, if there is no true spatial effect. Now, doing this in this kind of permutation scheme is fairly time consuming. So in 2012 we actually put out a paper that shows that we can solve that analytically, which is much faster, but the concept is still the same. In the actual ALE analysis, we combine the values at a particular location. Our null hypothesis says location doesn't matter, so we refer against a null distribution where we have the combinations of randomly picked voxels. And if we do that, now I'll show you the whole cascade. So we have a set of experiments with all the activation foci, replace them by the Gaussians to get the activation likelihood, and then threshold them. And these are the areas that show a significant convergence of the coordinates, and this fits actually sort of as a phase validation fairly well with some newer data that we acquired. Any questions on that? This is the basic principle of a neuroimaging meta-analysis. You take the foci, you model the uncertainty, you compute the union, so you integrate across the individual experiments, and you test against a null distribution reflecting random spatial association.
Sorry, does it mean that your null hypothesis strongly depends on the reported foci?
In the way that you estimate the null distribution from the data, yes. So in your test statistics, so to speak, you compute at each location. Let's say you have 30 experiments, and so your true analysis looks at the convergence across the 30 experiments at a particular location. And your null says basically location doesn't matter. So what kind of values would I see if I pick from just somewhere? So this also enforces what I said earlier. We are not looking at any effect sizes, we are only asking spatial questions. So I test where is the convergence higher than what I would see if there was no spatial meaning in the data.
Another question, do you weigh the individual studies, for example, according to the number of participants in each study?
Yes, not for example, but actually for the number of participants. And this actually influences the spatial uncertainty. So when I go a little bit deeper into the uncertainty estimation, There are two factors to it. One is what you could call the inter-template or inter-laboratory variance. It's basically spatial uncertainty that can be attributed to the fact that you can use different normalization strategies, templates, and so on. And this is kind of like a fixed baseline level of uncertainty. And the second part is the between subject variance. So if you would have a huge number of subjects, then basically you could be fairly sure that your group peak is the true population peak. If you have, however, let's say just 6 or 7 subjects, then you obviously have a random sampling from the population, so your group peak will vary somewhat around the population peak. And this kind of varying around the population peak is less if your subject number is higher. So there's, let's say, an indirect weighting by the fact that larger studies have a smaller Gaussian, so more certain localization, and hence a higher localizing power. So that's the only weighting. We have already, we and many other people, tried to play with tons of other weightings, like for what kind of statistical threshold did they use, what kind of height t-value did they report, and so on. In the end, to be honest, it all doesn't help. Many things are inconsistently reported, and if you use something like significance level or something like this, then you do get at times into the inverse problem that if you have a small group, just by chance you can actually get the most significant effects, whereas in a large group the effects often are even a little bit less. So playing around with a lot of different ways of weighting, what it came down to is weighting by sample size through uncertainty is very helpful. Anything else gives you a lot of free parameters you can fiddle with, but doesn't really help things. And so I was always for less things that the user can set. You had a question as well?
Yeah, just wondering, does it seem to be totally independent of like publication bias or something like that? There's no apparent convergence on a particular area over time.
Well, it's actually fairly interesting that it's not independent of publication bias, but it seems fairly independent of it, given that for whatever reason, most neuroimaging stuff gets published sooner or later. This is an, let's say, sociologically very interesting aspect, that neuroimaging seems to have much less publication bias in other fields. Now, why is that? Suppose you pay $1,000 for each hour scan time. So you've invested a lot of time and money into a study. Even if it was crap, and you didn't really find anything, you can just not afford not to publish it. So you see a lot of fMRI studies, really crappy ones, being published in low-level journals, which, from a meta-analytic perspective, is great, because they're all in the literature. So it seems that, in particular, my primary comparison is always cognitive psychology. I think there are lots and lots of behavioral studies, sort of summer intern projects that never get published, but then again, we didn't invest a lot in that. But given how much a lab has invested into an fMRI investigation, it's gonna get published sooner or later somewhere. Now, but we have a different publication bias that is, however, not too difficult to deal with, which a lot of people use a priori regions of interest or small volume corrections on. If you did a study on face processing and you do not find the amygdala, then reviewers will give you a hard time. So you just place, let's say, a well-positioned sphere in the amygdala. If it's small enough, you get something significant there, and you can publish it. Now the key thing is, for meta-analysis, we can only use whole brain analysis. Anything which is reported for a priori region of interest, small volume corrected data, no. That is a self-fulfilling prophecy then. If we take all the small volume corrected stuff that people report, then it's kind of like going in circles and circles and circles. We can only take things from whole brain analysis. And then we don't get around all the biases.
I agree with you. You said in your follow-up study that this fairly well correlates with the meta-analysis, and I agree with that on the areas of convergence seen in the upper panel. But I see additional areas of convergence in your follow-up study.
Here it was just, this was the visual pacing.
So my question is, how are you treating this information?
Well, in fact, this is kind of like, let's say, I include that usually as a sort of phase validity comparison, but it actually illustrates very nicely the point I made earlier, that you have study-specific peculiarities that modulate what you find. So in this particular study, the finger tapping was visually paced, so there was a blinking circle that indicated the subjects when to press. And this is exactly the activation related to the visual pacemaker. A lot of the studies here did not use that. So this actually illustrates very nicely what I said earlier, that how exactly you do the experimental design will influence your results. And this is exactly what we see here. Now, this is fairly straightforward. You can also do that in a little bit more complex fashion. And there we come to somewhat of like a meta-analytic decomposition of more complex field. Now, finger tapping was almost as simple as you can get. Now let's get to something else that is probably more interesting but also more complex, working memory. Huge field, lots of interest from psychologists, clinicians, neuroimagers. And what you see here is the activation findings from about 200 working memory experiments. Now what's the first thing you note? Which brain area is not associated with working memory? Oops, gets difficult here, right? So this is really one thing that, again, illustrates what I said earlier, is there are so many different factors, and each influence kind of where things happen, that it's almost impossible not to get any activation somewhere. If we compute the activation likelihood estimate, then it basically reflects that. We see areas of fairly high convergence, but there's basically no area of no convergence. But if we compare that against the empirical null distribution, we can see that it's basically a frontal-parietal network spanning the inferior frontal sulcus and the premotor cortex. Then here the anterior insular and Broca's region, plus the intraparietal sulcus on both sides. That's kind of like the core network for working memory processing. So that's the main effect. That's the convergence across all of these 200 experiments.
I think that's particularly interesting because
Yes.
Adrian Owen's task has been much used for working memory, the Alpbach task. And I suspect
Can I go one slide further?
Okay, yeah.
I hope it's what I have on the next slide. So basically, what we can do now. So I'm finished with that sentence. I hope because I have two different examples for what could be on the next slide. I hope it's the right one. So that's the main effect. Basically, looking for where is the convergence across this huge heterogeneous pool of studies. Now we can also sub-differentiate and use what we call meta-analytic contrast. So the question is, where is the convergence for set A higher than for set B? Or is a group of studies A more likely to result in activation at this voxel than the group of studies B? No, it's the right slide. So null hypothesis is labeled exchangeability, and here you see a comparison of the two most widely used working memory tasks. The Alpbach task, where you basically see successive stimuli, and you have to say whether the current one is the same as the previous or the same as 2 back. And the Sternberg task, so here you have basically a constant cycling. Each stimulus is probe and material for the future. The Sternberg task is the other very popular working memory task. You see a set of items, let's say 5 letters, there's a little break, and then you see one letter and you ask, was that one of those that you memorized? Now the big difference is, the Alpbach, as I said, has a lot of maintenance and cycling, The Sternberg is a more simple input-output task. And now what we did here is contrast these on the meta-analytic level. So the fact that there are many different versions of the NBAC, what kind of stimuli, what kind of timings and so on, a lot of different Sternbergs, again, stimuli, timing and so on. It's all, this should cancel out. And now we're looking at, like, on this broader overarching level, are there consistent differences? And yes, indeed there are. As you can see, in red here are areas that are more likely to be recruited by the NBAC task, and quite strikingly, you see here on the prefrontal cortex a lot of red. Now this is really crucial in a way. Think about clinical application. You want to do an fMRI study, working memory in schizophrenia, and you ask your colleague, well, do you have a task that can use in the scanner? Now, if that happens to be a Sternberg task, you a priori have a much worse chance of writing a great paper about prefrontal hypoactivation, because maybe your task doesn't even recruit the prefrontal cortex. So it really means that the choice of your task can already bias your result, and on the other way, that with this kind of meta-analytic decomposition, we can now take a broader construct as working memory and really look at different aspects. It's like, where are these kind of tasks more likely to evoke activation than these kind of tasks? And we can do that now on a level that is way beyond what you can do in a single study. In a single study, you can maybe compare two tasks with each other. Here we can basically compare whatever we want with each other. We can also look at things like task set effects, so doing a task versus not doing it, and load effects, more difficult versus an easy version. And quite interesting, you see again something here in the prefrontal cortex, left anterior prefrontal. This seems fairly specific to task set, so doing it versus not doing it. Whereas here on the more posterior frontal cortex, this seems to be much more load related, so more likely to increase activation in difficult versus easy tasks. So with these kind of approaches, you can now take broader constructs, working memory, attention, motor control, executive functions, and if you want, deconstruct it and test what are actually significant factors among which study results vary. So, in other words, we can actually now look at properties of neuroimaging studies. So not properties of a task, but rather, the brother said, like, if you do the tasks this way, what kind of things do you have to expect? Together, this is the one major application. There's another very interesting application to these kind of neuroimaging meta-analysis database techniques, and that's the problem of functional inference. Now, I guess the following slide will not be as appealing to you than it is to many people in neuroimaging. This is Broca's region. Who of you has heard of Broca's region? Yeah, that's kind of the problem. It comes across better if you talk to a neuroimaging audience. Now, I'm sort of getting ahead of myself now. Broca's region is the speech region in the brain. It's the language center, the very first functionally specialized region described in the brain at all by Paul Broca in the mid-19th century. And so Broca's region is language, the language area. Now the thing is, however, there are a lot of other things that activate that area as well. So what that means is, illustrated on a very simple example, if you have a particular part of the brain, a particular region, and if you want to interpret that, no matter how you want to interpret it, you will always find one or two studies that localize a particular thing to this region. And this is the problem of functional inference. Basically, if you have a particular area, you cannot really subjectively say what does it do. Even for something as straightforward as Broca, there's a lot of variance. And it gets much worse for other areas. So really what we want is to compare it to the overall neuroimaging work. So this part of the brain, what has that been implicated in? And we can do that by the emergent neuroimaging databases. There are two big databases, BrainMap and Neurosynth. And the key idea behind this database is the following. You collect as much neuroimaging experiments as possible. And for each experiment, you store X, Y, Z. Where are the activations? And you code in some way what has been done in that experiment. I'm saying a bit loose here because there are major differences between the databases, how they do that. But the key message is you have neuroimaging databases like BrainMap that store for thousands and thousands of experiments where are the X, Y, Z coordinates for the activations and what did they do. Now if you have that, you can actually do a quantitative inference on what's going on. Suppose we take this cluster here in the primary motor cortex. Now we can ask, what is the likelihood of activation in this area given a particular task? So were experiments using a particular task more likely to activate its ROI by chance? And if we test that and show you only the significant ones, you see that it's exactly as one would expect from the motor cortex. So it's strongly associated with action execution, and in terms of paradigms, finger tapping, grasping, pointing, so anything motor related. Now here it was simple. And this is why I use it as a toy example. Primary motor cortex movement. But it shows you that we can statistically answer the question, what is this part of the brain doing? And we can also do that with more formal reverse inference. So if we see an activation here, what's the probability of a certain task being the reason for that? And again, we come to the same conclusions. Action, finger tapping, grasping, and so on. So, in a nutshell, we can do two things with neuroimaging meta-analysis. One is that we can ask where is there convergence over experiments probing a particular construct. And we can, in addition, also test whether there is a differentiation given, for example, what kind of task we use. We can also go the other way and say we have a particular region in the brain, and I want to know what is this part of the brain doing. And I want to do that, get a statistical, objective answer to that question. And we can do that by reference to these kind of neuroimaging databases, where we can then formally test which kind of tasks or behaviors are associated with activation in that particular region. Moreover, we can also even look at functional connectivity with these kind of approaches. Now, I'm using the same seed region again, here the primary motor cortex. And remember here, I pulled out all study in the database activating that region. And I looked at their description. Yeah? Using this as a mask, pull out all studies that activate there, and look at the descriptors. What did they do? Now what I did here is to have the same studies, but now plot all the activation foci. Of course, we have the highest convergence here in the seed. That's what we defined the studies by. But you see, there is also a lot of other activations. And in fact, there are about 2,500 activation foci reported in these 168 experiments. Now we have to point out again, experiments are only identified by the fact that they feature activation here in the seed region. And now we can again use activation likelihood estimation over these experiments. And what this gives us now is for each voxel a probability. How likely does this voxel co-activate with our seed region? So a probabilistic representation of co-activation. And we can again threshold this, and we basically get those regions that are significantly co-activated with the seed. So whenever the seed is active, which other regions are active as well? And this gives us a window into task-based functional cognitivity. And it's really interesting to compare that with, don't have that, I have that here, but I think in the interest of time I'm going to work that very briefly. This is actually really interesting because we have resting state cognitivity. That's basically functional cognitivity in the absence of a given task. And we have this kind of co-activation mapping. That's functional cognitivity while certain tasks are being done. And yeah.
Could you just give us 2 or 3 sentences on thresholding and what you think about how it should be used? Because it seems that it can be used to have things appear or disappear or whatever.
Yeah. Well, it depends what your perspective really is. So I personally am often actually interested in the unthresholded maps because it kind of gives you like the whole brain pattern of things. It does have obviously the disadvantage that you cannot make any easy statements about this is significant, this is co-activated or not. So the standard would be, and I think you can't really get around it in the current field, is to use significant thresholding corrected for multiple comparisons across the whole brain. So while I personally like a lot of arguments for not doing it, as you said, you can make things disappear and so on, things that are not whole brain corrected significant, you will have a fairly difficult time publishing. So it's just the state of the field at the moment. And this is just an example here for the anterior insula. So there was a study that we defined in conjunction with a working memory meta-analysis, the core of working memory, and schizophrenia atrophy. We got one region here that was both sort of in the working memory core and atrophic in schizophrenia. We can have a look at what this region is doing in terms of different behaviors. It's mainly language and cognition. Same as if you look at the paradigms that activate here, associate recall, Sternberg, word generation. It's basically language and higher cognition. Now the interesting thing is now we can do this meta-analytic co-activation mapping, resting state connectivity, and also structural covariance. So we have a lot of different approaches of how we can identify which regions is this one interacting to. And the key thing is here they reflect very different things. Mechanism is really in the presence of this kind of externally structured tasks. If this region activates, which other as well? Resting state looks at this endogenously controlled, unconstrained flow of thought. Which regions co-vary in that state with my seed region? And structural covariance really looks at kind of like the long-term plasticity effects. If you are somebody who has a large region here, which other regions are large? And if your region here is small, which others are small? So which across subjects, which other regions correlate in their size with the seed? Now, the interesting thing is you can then define a conjunction across all of them. And the nice thing here is all of these approaches have their own problems associated with them. They all have their methodological pros and cons, their drawbacks, their limitations. But importantly, they are completely independent. This has nothing to do with this, has nothing to do with this. Now, if you do, and they are different in terms of subjects, and different in terms of methods, and different in terms of what they look for. So, here now looking at the conjunction, these are the regions that seem to be really robustly interacting with my seed. No matter how I look at brain connectivity networks, they always come up together with the seed region. And These are again associated with language, attention, working memory, inhibition, and sort of like executive functions. Now I skip this. Just want to point out one other thing. You can also look at the differences. So, same seed region, but look at like, what is specific to each part of connectivity. So now we're really looking at the properties of these connectivity measures. And then we see that in the resting state, in red here, this region is particularly coupled with regions that are involved in sensory monitoring and reward. For co-activations, really cognitive processes, and quite interesting, structural covariance, emotion and social cognition. So we can formulate some ideas that resting state connectivity may be quite strongly related to inner milieu monitoring and saliency. Co-activations really reflect the brain's role as an input-output machinery, and structural covariance may be something more related to what I tentatively called real-life wiring. Because remember one thing, those things that we do in the fMRI scanner are fairly different from things that we do in everyday life. So connectivity may also to a large degree reflect state aspects. Coming to the conclusion that there isn't really such thing as brain connectivity, but rather, well, just in function connectivity, but rather it seems depending on how we look at things, the answers to large-scale brain connectivity can be fairly different. And I think this is of tremendous interest in light of things going on like the Human Connectome Project in the US, but also the Human Brain Project, where many people just straightforwardly talk about the connectome, the connections in the brain. Now, this is a hyper simple example. We just look at one particular small seed region. Yep, just this one little region, the anterior insula. And we just look at three particular ways of looking at connectivity. And they are widely used, but by far not everything that we can do. And still you see, yes, there is some convergence, but you also see marked differences between the approaches and what kind of connectivity they reveal. What we don't really know is how much of that may be different noise properties, different biases, or I'm slightly leaning towards the second, this is just different aspects of connectivity. We're looking at different things. Whether you are just lying there in the scanner, let your mind wander, whether you're doing a particular cognitive task, or whether we look at which kind of regions grew together over the last years and decades. It's just completely different things that we look at. And they give you different answers to what kind of regions is the seed connected to. Now, I'm going to skip the last part. I think the slides get uploaded. But I think in the interest of time, I'm not going to talk about that anymore. But rather come to my summary. So I hope I have shown you that the insight from each individual neuroimaging study is fairly limited. But the high degree of standardization allows pooling of the results and hence inference on converging evidence. Coordinate-based meta-analysis provide a statistical tool for the objective integration of findings, which then allows also to look at the effects of, for example, experimental tasks. Database-driven functional decoding allows objective forward and reverse inference, and hence answering the question, what is this part of the brain actually doing? Meta-analytic connectivity modeling offers a new approach to task-based functional connectivity. And with that, we now have at least 4 different ways to define brain connectivity: co-activations, resting state, structural covariance, and I haven't talked about that, structural connectivity in the sense of fiber tracking. So this leads to a new field which is just emerging which is the comparison of different connectivity measures. And finally, this is what I have not talked about, co-activation-based oscillation allows you to identify cortical modules and map the brain. With that, I would like to thank my group, former people in my group and the collaborators, and obviously thank you for the attention and discussion.